Hello everyone, we're going to go ahead and continue on in the 4.1 notes. In this video we're going to look at topic 2, which is going to focus in on signal transduction pathways and how that's going to ultimately lead to different cell responses. And so in topic 1, just to remind you guys, we were looking at what happens when a cell receives a signal. And so there's these ligands, these signaling molecules that bind to specific receptors. And that's what we looked at in topic 1. In topic 2, we're going to look at this next part here, which is a whole sequence of events now that are going to take place inside the cell once that receptor protein becomes activated after binding to a ligand, which is ultimately going to lead to some kind of response in the end. So that's what's going to happen here in topic 2. I will go ahead and warn you guys that it is some of the things that we're going to look at here in these signal transduction pathways that are kind of tricky to understand at first. But hopefully, if you're paying close attention, it will make a lot of sense. And we'll try to go over the main ideas here. But basically, once that ligand binds to a receptor protein, it's going to trigger a whole sequence of events now taking place inside the cell. And that sequence of events taking place inside the cell that's going to ultimately lead to a certain cell response is what we call a signal transduction pathway. And that pathway consists of a whole series of steps, which really, by series of steps, I mean mainly it's usually going to involve different proteins becoming activated in the cell in sequential order. And so you have the receptor protein, that's the first protein becoming activated. That's going to activate another protein, and that protein is going to activate another protein, and then those proteins are going to activate other proteins in the cell until finally you activate certain proteins in the cell that are going to now actually do something, a specific thing that the cell is supposed to do at that point. So there are a lot of steps. It's common for these pathways to have a lot of steps. Now, actually having a lot of steps has a couple benefits. One benefit is that it allows the signal to be amplified. And so if we're just starting with, like, let's just say one ligand molecule binding to one receptor on the cell, which is pretty insignificant because a cell is gigantic compared to these proteins that we're talking about. A cell would have thousands or millions of receptor proteins. And we're just talking about one signaling molecule binding to one receptor protein on the cell. Well, in these signal transduction pathways, what's going to happen is we're going to see proteins becoming activating other proteins. And every time that proteins are activating other proteins and those proteins are activating other proteins, we're going to see an exponential increase in the number of proteins becoming activated. So by the time that we get to the protein inside the cell that's being activated, that's actually doing something, causing the cell to respond, we're going to be having thousands, millions of those proteins now activated in the cell a lot of the times, starting with just one receptor protein becoming activated. And so that will hopefully make sense by the end of this video. Another thing that having multiple steps, or another reason having multiple steps is beneficial, is that it allows kind of for more intricacy in terms of the possible things that could now happen in the cell when a ligand binds to a cell. So when a ligand binds to a cell, since there's multiple steps that can take place, it's going to be a different, unique set of steps depending on the ligand involved and the receptor protein binding to that, that's binding to that ligand. That's going to then trigger a certain set of steps to now take place inside the cell. And that's going to be unique and different depending on the ligand and depending on the receptor protein that it's binding to and depending on the cell inside the organism. We'll look at, in these notes, we'll even talk about how sometimes the two different cells in your body can be responding to the same signaling molecule, the same ligand. The same ligand can bind to one type of cell in your body and also bind to another type of cell in your body. But then those two cells can have completely different responses to that same signaling molecule. And that's because there's different steps involved, which is going to lead to a huge possible number of outcomes in terms of what the response is going to be. And so that's the overall main ideas of some of the things we're going to look at. Some common components of these signal transduction pathways that we're going to dive into right now are phosphorylation cascades and secondary messengers. So a lot of times in these sequence of events taking place inside the cell, there's going to be phosphorylation cascades, which I'll show you in a moment, and secondary messengers, which I'll also show you in a moment. that are involved in these pathways. So let's start with phosphorylation cascades. This is one common thing that we see in a signal transduction pathway. And so basically, a lot of times what's going to happen in the cell is proteins are going to become phosphorylated, meaning that certain proteins, there's going to be phosphate groups that are put on certain proteins. Certain proteins are going to have phosphate groups attached to them. And when they have phosphate groups attached to them, they change shape. So anytime something binds to a protein, this is a common theme we've seen all year. When things bind to proteins, whether it's enzymes or other proteins or whatever it is, they're gonna change conformation, change shape, and sometimes they change into their active shape, where they now start doing their job, and then sometimes they change into their inactive shape, where now they're not doing their job. And phosphorylation is one common way that takes place. When a protein becomes phosphorylated, when a phosphate group gets attached to it, a lot of times it changes shape into its active conformation. So for some proteins, this is how they turn on, is there's a phosphate group that gets attached to them. And then sooner or later, that phosphate group gets detached from it, gets removed from that protein, and then the protein changes back into its inactive conformation, and then it's not doing anything. So it's kind of like the on and off switch for a protein by adding these phosphate groups. And so this is a common thing that happens in these signal transduction pathways, is you see proteins becoming phosphorylated, changing their activity. And so there's a type of protein whose job is to phosphorylate other proteins. And so the name of a protein so if you have a protein and his job is to find a specific type of protein in the cell and put a phosphate group on it, we call that a kinase protein. So a protein kinase is a type of protein whose job is to put phosphates on they put phosphate groups on other proteins. They phosphorylate other proteins. And so if I was a protein kinase, my job would be to go find a certain protein inside the cell. There's a certain type of protein that I'm looking for. And then I grab it, and then I'm going to put a phosphate group on it. Like, that's my job. And then I go find another one, and I grab it, and I put a phosphate group on it. Like, that's what a protein kinase's job is to put a phosphate on a certain type of protein. Okay. So in general, there's a lot of different proteins whose job is that. And so in general, we call them kinases. So kinases is just the general term for a protein whose job is to put phosphates on other proteins, to phosphorylate other proteins. Now, the way they do that is they take a phosphate from ATP. If you guys remember, ATP is this high energy molecule that has 3 phosphates, and that third phosphate is very removable. And so what a kinase does is a kinase will go find an ATP molecule and take the phosphate from ATP and go put it on a different protein. Like, that's what he does. And then he goes and finds another ATP molecule. He's going to take the phosphate and then transfer that phosphate to another protein. That's specifically what a kinase protein is doing. And what we see in these signal transduction pathways is we see a whole cascade of phosphorylations taking place, accomplished by a series of protein kinases who are phosphorylating other protein kinases who are phosphorylating other protein kinases. So we call that a phosphorylation cascade. So let me kind of explain what that looks like here in this picture. So if you guys look here, there's a protein kinase. We'll call it protein kinase 1, a certain type of protein kinase who's becoming activated inside the cell. And that happened after the receptor protein became activated. Eventually, this guy becomes activated, this protein kinase. He becomes activated, and his job, like I said earlier, is to go find a certain protein and phosphorylate it. So his job, if you guys look at this picture, is to go find protein kinase 2, a different protein kinase, and take an ATP and put a phosphate on protein kinase 2. So what this guy is doing is he's taking a phosphate group from ATP, and he's taking that phosphate group and putting it on protein kinase 2. So then you end up with protein kinase 2 now having this phosphate group, and the ATP no longer having that phosphate group. That's what this guy does. That's the chemical reaction that he catalyzes. He transfers the phosphate from ATP to this protein kinase. Now this protein kinase 2, when it becomes phosphorylated, you can see it even changes shape here, and now it's in its active shape. And then his job is to, again, go find another specific protein, this protein here, and his job is to go find these proteins and phosphorylate them by, again, what he does is he takes a phosphate from ATP and puts it on this protein, which now this protein will be phosphorylated and changes shape, and now it's in its active shape. And so you could have a series of protein kinases who are phosphorylating other protein kinases, which is going to lead to a sequence of phosphorylations taking place inside the cell, which we call a phosphorylation cascade. This is just a common type of thing that we see in a signal transduction pathway. And then we'll kind of talk about this more throughout the video, but just to point out here, just when these protein kinases become phosphorylated, like this guy right here, just notice that he's a common misconception. Students think that he's going to take his phosphate group and put it on this next guy, which is not what he's doing. He's not giving up his phosphate group. He's finding ATP. His job, a kinase protein's job, is to find ATP and take a phosphate from ATP, not his phosphate. He keeps his phosphate, and he is going to put a phosphate on a different protein, which is what he's doing right here. That's his job right here, is to do this. Now, eventually, these phosphate groups that are being added to these proteins, they're going to be removed. There are enzymes called phosphatases. So protein phosphatases are proteins whose job is to remove those phosphates. And in that picture, it's represented by this little guy right here that says PP. That's a protein phosphatase. And this is a protein whose job is to catalyze the reaction that removes the phosphate from this guy. So you guys can see that his job is to find this guy and take his phosphate off. And so then this phosphate is removed and he goes back to his inactive shape until another protein kinase phosphorylates him again. So just know these things aren't permanent, I guess is what I'm trying to say. These proteins are becoming phosphorylated with these phosphate groups, but those phosphate groups will be removed eventually. And it's not just this permanent thing that's going to take place now forever inside the cell. This is just a temporary thing now that the signaling molecule is binding to the receptor protein. Anyway, hopefully that kind of makes sense so far here, what a phosphorylation cascade is. Another common feature that we see in the signal transduction pathway are secondary messengers. Secondary messengers, well, okay, so the ligand, we consider the ligand as the primary messenger or the first messenger, right? And usually the ligand is outside the cell, and usually the ligand doesn't go into the cell. A lot of times these ligands are binding to membrane receptors. But then in the sequence of events that take place after the ligand binds to that receptor, a lot of times it leads to a secondary messenger being released now inside the cell. So a secondary messenger is a small polar molecule that can quickly and easily diffuse inside the cell that gets produced on the interior side of the cell and then will diffuse throughout the whole cell and then go and usually bind to other proteins to activate them. That's what secondary messengers do. And so that's the job of a secondary messenger. It's basically to be the signaling molecule now inside the cell. And usually thousands of these get made and then they diffuse quickly throughout the cell to go turn on different proteins, which is what's happening here. Now there's two common, the most common example of a secondary messenger is a molecule called cyclic AMP. So cyclic AMP, or a lot of times for short, we call it CAMP. This is probably one of the most popular secondary messengers that we see in the signal transduction pathways. And there's a protein who makes CAMP, cyclic AMP, the secondary messenger, and his name is called adenylyl cyclase. That's the name of this enzyme whose job is to, basically what he does is he actually takes ATP and he turns it into CAMP. And then CAMP is the small molecule that can quickly diffuse throughout the whole cell. And so that's his job, is to produce the secondary messenger. And you guys can see in this little picture here, it's kind of connecting a lot of different things that we've talked about. Here's a G protein coupled receptor and the ligand, the primary messenger, who's going to bind to that G protein linked receptor. And then that G protein linked receptor is then going to activate the G protein by allowing a GTP to be put on this G protein, if you guys remember that from the first video. And then that G protein now is activated, and it's going to go and bind to another protein on the cell membrane. In this case, it's adenylyl cyclase, who is now going to become activated once this G protein binds to it. And then his job is to take ATP and turn it into CAMP, and then he's going to make a bunch of CAMP, and that CAMP is going to spread out throughout the whole cell. And then you can see that the CAMP can then bind to protein kinase A, a certain type of protein kinase inside the cell, and then activate that protein kinase. And then that's probably going to start a phosphorylation cascade where that protein kinase is now going to phosphorylate other protein kinases who are going to phosphorylate other protein kinases who are going to end up phosphorylating some protein in the end. And then you finally get to your cellular response. So this take a moment to like look at this picture here, to kind of start connecting a lot of different and reviewing a lot of different things that we've talked about so far. So that's CAMP, okay, very common secondary messenger. Another common, there's a lot of different examples of secondary messengers, but another common example is calcium ions. Calcium ions can also act as secondary messengers. You guys can see in this example here, there's a series of events taking place that lead to a calcium ion channel opening that releases calcium into the cell. And then those calcium ions, those are small polar ions that can very quickly diffuse throughout the whole cell, and then they can go bind to specific proteins and activate those proteins, who then continue on the message to other proteins. These are really important actually in your muscle cells. I talked about that in the first video actually. And in order to get cells to divide, these calcium ions, they bind to specific proteins involved in muscle contraction, or in proteins that are needed for cell division. But anyway, those are secondary messengers. And so that's another common thing that we see in these signal transduction pathways. So hopefully that vaguely makes sense so far. So in these signal transduction pathways, in these sequence of events that take place inside the cell, that are initiated by a ligand binding to a receptor, some of them are phosphorylation cascades. That's a common thing that we see happening, which are these protein kinases becoming activated and phosphorylating other protein kinases who become activated, and so on and so on, and secondary messengers getting released inside the cell. And so just try to piece that together in your head, that these are just common players we see happening in these signal transduction pathways. Now, like I said at the beginning of this video, one benefit of this, it seems kind of complicated. Why does it have to be so complicated? And like many things in biology, there's a reason that it's complicated. One reason is that by having all these different steps involved, it allows the signal to be amplified. So you can start with just one signaling molecule binding to the cell, and this kind of table here kind of shows you and keeps track of how many molecules are being activated. So if we start with like one protein receptor being activated by a signaling molecule, just one of them, which is like the most insignificant thing in the world for a cell. That's like in the city of Fresno, one person getting hired for a new job. Like it's nothing. Cells are really big. And we start with one protein who's now activated. He's like going to turn on today. He's going to start to work and start doing his job. And what happens though is he's going to then, in this case, they're talking about how this is a G protein link receptor, and how that G protein link receptor is now going to activate some G proteins. And he's going to activate, and according to this, it says he's going to activate 100 G proteins. So now we have 100 G proteins that are activated. And then those 100 G proteins that are activated are going to go activate another protein. In this case, it's the adenyl cyclase, which I showed you earlier. That's the guy who makes CAMP. Right here, this secondary messenger, this protein right here, adenyl cyclase. So there's going to be 100 G proteins, and each of those G proteins are going to now go and activate these adenyl cyclase enzymes. And this guy can bind to this one, and then another one can bind to another one, and the other one can bind to the other one. And then these guys are going to make CAMP. And then each of them can make lots of CAMP while they're activated. And so if you keep track of what's here, so then adenyl cyclase is going to take ATP molecules and turn it into CAMP, the secondary messenger. And now we have 1,000 CAMP molecules who are going to spread out and diffuse inside the cell. And those 1,000 CAMP molecules are then going to go bind to and activate some protein kinases. Starting with protein kinase A, they're going to bind to that protein kinase and activate it. And then that active protein kinase A, there's now, this is 10,000. There's now 10,000 of them activated. And each of those protein kinase A's, there's 10,000s of them now awake inside the cell. They're going to go phosphorylate another protein kinase. So then they go and find another kinase protein to activate. And each of them activates a handful of these other protein kinases. And now there's 100,000 of these guys activated, and then those protein kinases then go find this protein here, glycogen phosphorylase, I don't know this enzyme, and then they activate this guy, but now each of them can do that a handful of times, and so now there's 1 million of these guys activated, and then those guys, these are enzymes whose job is to take glycogen and break it down into glucose molecules inside the cell, and they can do that many different times before they become inactivated, and now you have what is this? I don't even know. 10 million, 100 million, 100 million glucose molecules being produced inside the cell that are going to be released from the cell into your blood. This is an example of like a liver cell, for example. So that's what it's saying right here. One molecule of epinephrine, a signaling molecule, can bind to a receptor on a liver cell, and then this signal transduction pathway and these series of steps, in each step we're activating more and more and more and more proteins, leading to eventually 1,000 different glucoses now being released into your blood, all starting with one signaling molecule. And that's what we mean by a cell response. We're talking about like the whole cell is now focused in on doing this big task of, in this case, releasing glucose into the blood. And so that's just know that that's one benefit of these signal transduction pathways, is that they hugely amplify a signal that's received by a cell to end up with a big cell response. It also allows the cell to have a specific response to a signal. And so depending on, this picture here is kind of showing you that depending on the proteins inside that are present inside the cell, so different types of cells, like this is showing you cell A, cell B, cell C, and cell D, different types of cells have different proteins that are going to be involved, that are in the cell, that are ready to be involved in a signal transduction pathway. And depending on what set of proteins you have available for your signal transduction pathways, that's going to determine what sequence of events takes place inside the cell and what response gets produced. And so you can see in these pictures here that some of these guys have different sets of proteins that are going to activate other sets of proteins, just depending on what's available in the cell that we're looking at, which is going to lead to different unique responses inside the cell. And so that's another benefit of these signal transduction pathways, is they allow cells to have specific responses to specific signaling molecules that are binding to specific receptors, which are going to produce a specific response depending on that type of cell. And so a good example, well, just one easy example, well, not an easy example, but a good example, I guess, to show you guys how this allows for some specificity, is liver and heart cells and how they can both respond to epinephrine. So epinephrine is a hormone, a signaling molecule that's released into your blood. And this hormone can bind to receptors on your heart and receptors on your liver cells. So your heart cells have receptors for epinephrine, and your liver cells also have receptors for epinephrine. However, the cool thing is that when epinephrine binds to your heart, it actually produces a different response in your heart cells than when epinephrine binds to your liver cells. So your liver and your heart cells are actually going to respond differently to the same signaling molecule being produced inside of your cell, inside of your body. So when epinephrine is released inside your body, it travels in your blood and it's going to go bind to your heart cells, and it causes heart cells to contract faster. So it activates motor proteins in your heart, causing it to contract and beat faster. Your heartbeat goes up because of epinephrine. And epinephrine can also bind to cells in your liver. And when it binds to receptors in your liver cells, it causes glucose to be released into your blood. So liver cells store glycogen, and then that was the example I was showing you earlier in this sequence of events here. When epinephrine binds, it leads to a bunch of glycogen breaking down into glucose, and that's going to be released into your blood. That's the response the liver has to epinephrine. So they both have two different responses. This guy is going to start contracting and beating faster. This guy is going to break down glycogen and release it into your blood, both to the same signaling molecule. And the reason that is a common AP question. Like, how is it possible that that two different cells can respond differently to the same signaling molecule? Like, they're both binding to epinephrine. And the answer is because liver cells and heart cells have different sets of proteins inside of their cells. It could be the receptor, the epinephrine receptor might be a little bit different, or maybe it's the same receptor, but it's then going to lead to a different transduction pathway, a different series of proteins being activated inside the liver cell versus those that are activated inside the heart cell. And so the heart cell is going to have a different unique signal transduction pathway that takes place, leading to that specific response, versus what happens in your liver cells, which contain a different set of proteins and a different unique signal transduction pathway that's going to lead to what the liver cell does. So it's because of the uniqueness and the difference in the proteins involved in the signal transduction pathways of the heart cells and liver cells. I hope this is kind of making sense, because I feel like I'm not doing the best explaining it right now, but in class we're gonna draw this out a little bit together. to hopefully make a lot more sense. But hopefully after watching this video, it's kind of piecing together in your head a little bit. And so, and here's a picture kind of like illustrating or summarizing what happens in a liver cell. It's really good to just practice dissecting these diagrams. Here's an epinephrine receptor and here's epinephrine. So here's an example of your liver cell. That has a receptor for epinephrine. It binds to it. It becomes activated. This leads to a sequence of events that leads to this guy becoming activated, adenylyl cyclase, which I've talked about a couple times already. He's the guy who takes ATP and turns it into CAMP. And so he's going to make a bunch of CAMP. The CAMP's going to diffuse throughout the cell. And you guys can see in this picture, CAMP is going to activate certain proteins that lead to these guys becoming activated. And so this is an enzyme called glycogen phosphorylase, who's going to be phosphorylated probably after a phosphorylation cascade. So in this picture, they're not showing you probably a sequence of protein kinases becoming phosphorylated, phosphorylating other protein kinases who are then phosphorylating this guy. And when this guy is phosphorylated, he turns into his active conformation. And when he turns into his active shape, his job, you can see in this picture, his job is to find glycogen that's in the liver cell and break it down into glucoses that can then be pumped into your blood or released into your blood, released out of the liver cell into your blood. And then the CAMP, in this same signal transduction pathway, CAMP can also bind to these enzymes or lead to the phosphorylation of these enzymes here called glycogen synthase. And you can see in this case, glycogen synthase, when it's phosphorylated, actually turns into its inactive shape. That's what they're telling you here, that when this guy gets phosphorylated, he becomes inactive, whereas when this guy gets phosphorylated, he becomes active. All of that might sound complicated, but that's just the way it is. Some proteins, they become, a lot of times, mainly they become activated when they're phosphorylated, but some proteins become inactivated when they're phosphorylated, like this guy. And his job, his name suggests that he synthesizes glycogen, so it suggests that his name is to build glycogen. And right now what's happening is he's turning off. So he's going to stop building glycogen, and then this guy's going to turn on, who's going to start breaking down glycogen into glucose, which makes total sense because right now we're not trying to build glycogen, we're trying to break it down. So turn these guys off, turn this guy on, which is all happening at the same time when this cell binds to epinephrine. And so the types of responses that a cell can have to these to signaling molecules and the way they respond to other cells in the environment, there's lots of different responses that cells are going to have. Just some over big picture examples. It could lead to certain ion channels opening inside the cell. And when ion channels open inside the cell, that can change the concentration of ions and the electrical potential across the cell. That can change the overall charge inside or outside the cell. That's super important for certain types of cells, like your muscle and your nerve cells. Those are really important for that, for those to function properly. When you change the concentration of ions, or you change the membrane potential across the cell by allowing ions to go in and out of nerve cells or muscle cells, that allows your nerve cells and your muscle cells to conduct impulses or to contract. It could also lead to genes being expressed differently inside of a cell. So in your cells, there's these things called transcription factors, and those are proteins who basically allow certain genes to turn on and off. And in a signaling pathway, you could have transcription factors that become activated or inactivated that lead to certain genes turning on or off, which are then going to change what type of enzymes and proteins are being made inside the cell, which is then going to change what the cell is doing at that time. And then lastly, another just vague example of how a cell could respond to a signaling molecule is it could lead to certain enzymes becoming inhibited or certain enzymes becoming activated, kind of like what we saw in this example. This enzyme is becoming activated, this enzyme is becoming inactivated, and that's going to lead to the cell now accomplishing certain things or not accomplishing other things because of which enzymes are becoming activated or inactivated. And so all these are just vague examples of how the multitude of ways that cells are responding to different signaling molecules. And so we talked about that already. The last thing I want to talk about in this topic 2 is how it would be really bad if something went wrong in these signal transduction pathways. And so you guys are supposed to, this is a common scenario that comes up on AP test, is how, what would happen if like something in the signal transduction pathway is not working or is being blocked, or there's some kind of toxin or some chemical messing it up, and you're supposed to be able to think through what result that would have inside the cell. And so just to give you some examples, actually in a lot of cases, like when you guys hear about poisons or toxins to our cells, those toxins or poisons, those are chemicals that a lot of times they are messing with signal transduction pathways. They are messing with certain proteins involved in signal transduction pathways. And once you mess up a cell's ability to respond to certain signals from other cells, that's going to really mess up the stability of your body and how your body is operating. And so one example is anthrax. Anthrax is a toxin produced by a certain type of bacteria, and anthrax will actually lead to CAMP levels inside of your cells increasing. So it causes CAMP levels to increase inside the cell. This toxin kind of, it has the same effect that adenylyl cyclase has. It's able to build and synthesize CAMP. And so this toxin causes CAMP to be overproduced inside of your cells, which then ruins the ability of a cell to respond properly to signaling molecules, because there's too much CAMP being produced, or there's CAMP being produced when there's not supposed to be CAMP being produced. So then your cells start doing weird things when they're not supposed to be doing those things, and it's bad. And that's why it's toxic and can kill you. It's going to mess up important cells and how they're functioning. Another good example, super common example that comes up on AP questions is diabetes. And this is the example that I'll end with in these notes. But I do want to walk you guys through this. I know this is becoming a long video, but I do need you guys to be super familiar with diabetes and how that's a great example of these signal transduction pathways being disrupted. And so just to explain to you guys these three situations, people who are not diabetic, this is what's supposed to happen in your guys' body, is when you guys have a lot of glucose in your blood. So out here's your blood, and you have a high blood sugar. You have a lot of glucose in your blood. This is like right after you eat a meal, you now have a lot of sugar that was digested and it's now in your blood. What's supposed to happen is your pancreas is supposed to release insulin, which is this hormone, this signaling molecule. This hormone is supposed to be released into your blood as well. So when you have a lot of glucose in your blood, you're supposed to have a lot of insulin in your blood, because your pancreas, which is an organ in your body, is supposed to release this insulin. And that insulin, what it does is it binds to certain receptors on certain cells. For example, your liver cells and your muscle cells, they have receptors for insulin, and they can bind to the insulin, and then they trigger, it's actually a complicated transduction pathway, but in this picture they're not showing you everything, but there's a series of steps that takes place then after this receptor binds to insulin, and then there's a whole signal transduction pathway that takes place that ultimately leads to these GLUT4 proteins becoming activated on the cell membrane, whose job is to move and pump glucose into the cell. So it's supposed to take the glucose from your blood and move it and transport it into your cells. It's supposed to help increase the amount of glucose going into your liver cells and into your fat cells and into your muscle cells. That's what's supposed to happen. So this is how a normal or a non-diabetic person operates, is they eat food, their blood sugar goes up, insulin is released into their blood, it binds to insulin receptors on certain cells in your body, which then lead to glucose being pumped into your guys' cells. And that's going to lower the amount of glucose in your blood. So your blood sugar went up, and then your body's going to respond in a way that causes the sugar in your blood to be removed and imported into the cells, so taken out of the blood and put inside of your cells, so that your cells can then use that glucose or store it for later. And then your blood sugar goes back down to a normal level. This is what's supposed to happen. Now, people with diabetes, they have a problem with that. Something's not working the way it's supposed to. So there's two types of diabetes. There's type 1 diabetes and type 2 diabetes. And type 1 diabetes, this is when the pancreas does not make insulin as well as it should be making insulin. So there's a problem with the pancreas producing insulin. So when your blood sugar goes up, like after eating food, your pancreas does not, if you have type 1 diabetes, the pancreas is not releasing enough insulin into the blood, and that's causing these insulin receptors to not be activated when they're supposed to be, which causes these GLUT4 transporters to not become activated when they're supposed to be, which causes glucose to not be transported into the cells of your liver and your muscles and your fat when they're supposed to be. So then all of a sudden you have a bunch of sugar in your blood, and then you still have a bunch of sugar in your blood. Your blood sugar doesn't go back down, and all that sugar stays in your blood, which is not good, because we need to store that glucose for later for your cells to use. It also, when you have really high blood sugar, it messes with, especially in your brain, it messes up your brain and could lead to something really bad. I don't know. I think thinking stroke, but for some reason I'm thinking I'm wrong. It's a stroke or something, a seizure or stroke, that's going to take place because of that. It's not good. So anyway, that's type 1 diabetes. So it's messing up this whole system here. Type 2 diabetes basically is the same problem, but for a different reason. And people with type 2 diabetes, they have their cells have acquired a resistance to insulin. So they make insulin just fine. So their pancreas pumps out a bunch of insulin just like a normal person that's non-diabetic. So there's a bunch of glucose in the blood. Their pancreas releases insulin into the blood. But the insulin, when it binds to the receptors don't become activated or have a hard time initiating the signal transduction pathway that's supposed to take place inside the cell. So the insulin is having a hard time binding to or activating these insulin receptors, and these insulin receptors are not correctly initiating signal transduction pathways inside the cell that are, again, leading to these GLUT4 transporters not becoming activated. So then the glucose doesn't get transported into the cells and it stays in the blood. So basically you end up with the same problem here, is these people now have really high blood sugar and it's not going back down. So that's the difference though. So in type 1 diabetes, the problem is they're not making insulin. In type 2 diabetes, they are making insulin, but their receptors are not responding correctly to that insulin because of a problem with those receptors, or the signal transduction pathways that are supposed to be initiated from those receptors. So anyway, those are common examples of how messing up these signal transduction pathways is not good and could lead to bad effects on the human body and in living organisms. Anyway, that concludes this long video. I think that's it. So thank you guys if you're stuck through that. I'll see you in class.